So today I am going to explain about topic of carbohydrates. So carbohydrates are widely distributed compounds in plants and animals. So these carbohydrates contain carbon, hydrogen and oxygen. So contain carbon, hydrogen and oxygen. The ratio of hydrogen and oxygen similar as that of water. That means 2:1 ratio. Then carbohydrates are technically known as a polyhydroxy, aldehydes and ketones. Aldehydes and ketones. Either hydrolysis of these carbohydrates will give the polyhydroxy aldehydes and ketones. Then carbohydrates are classified based on the different aspects. So first one, based on the reducing property, these carbohydrates are classified into reducing sugars and non-reducing sugars. So generally carbohydrates also known as a saccharides. So saccharide is nothing but of a sugar. So these sugars synthesize in plants through the process of photosynthesis. Then this sugar stored in the form of sucrose, then starch, then other forms of the carbohydrates, then other forms. So first one, reducing sugars and non-reducing sugars. So reducing sugar, then non-reducing sugars. So carbohydrates are nothing but sugars. So first one coming to reducing sugars. So these reducing sugars generally contain free aldehyde and ketone groups. So free aldehyde and ketone groups are present in this reducing sugars. Due to this contain free aldehyde and ketone groups, if we add any chemical solution like a Fehling solution and Benedicta solution. If we add some Fehling and Benedicta solution to these carbohydrates by the gentle heating, these carbohydrates are reduced. That means this Fehling and Benedict solution contains Cu2 plus ions. That means cupric ions. These cupric ions converted into Q plus ions. That means Cu plus ions. So reduced cupric to Q plus ions. So generally this Benedict and Fehling solution blue in color. So generally blue in color, then alkaline solution. So blue color, then alkaline solution. By gentle heating, this blue color solution, when cupric ions are reduced to cuprous ions, this blue color solution, high pH solution converted into green color, yellow color, pink to reddish. That means this reddish color. So these type of sugars is known as a reducing sugars. So generally reducing sugars contain free aldehyde and ketones. So when or if we add some Fehling and Benedict solution to these carbohydrates, reducing sugars. So cupric ions, Cu2 plus ions reducing to Cu plus ions. That means Q plus ions. So this alkaline blue color solution change into green color, yellow color, orange to brick red in color. So this type of sugars is known as a reducing sugars. So best example, all monosaccharides. All monosaccharides, then glucose, then lactose, then maltose. So these are the examples of reducing sugars. Then non-reducing sugars. So in non-reducing sugars, these are not have this free aldehyde and ketone groups. So not having aldehyde and ketone groups. So due to these sugars cannot reduce the swelling and Benedicta solution. That means cannot reduce cupric ions to cuprous ions. So this is known as a non-reducing sugars. So best example, sucrose is a best example for non-reducing sugars. Then based on the hydrolysis, so hydrolysis will give the hydrolysis products. So based on the hydrolysis, then what type of products are producing by the process of hydrolysis? These carbohydrates are classified into three classes. So first one, monosaccharides. Then second one, oligosaccharides. Then third one, polysaccharides. So based on the hydrolysis and products of hydrolysis, carbohydrates are classified into monosaccharides, oligosaccharides and polysaccharides. So first one coming to monosaccharides. So monosaccharides are simple carbohydrates. So these are water soluble condition. So these are simple carbohydrates. So simple complex of structure is present. That means not have the complex of structure, simple structure, simple molecular conformation is present. Then low molecular weight. So molecular weight also very low in condition, water soluble in condition, sweet in a taste. Sweet in taste. So monosaccharides are simple, then simple or small molecular conformation, molecular structure, low molecular weight, water soluble, then sweet in a taste condition. Then these monosaccharides cannot hydrolysis further into smaller subunits. Why? Because these are the simple carbohydrates. That's why this cannot further hydrolysis into smaller subunits. Then these monosaccharides generally not dissolve, not soluble in organic solvents like ether, but these are partially soluble in alcohol. So partially soluble in alcohol but not soluble ether and other organic solvents. These monosaccharides contain 3 to 7 carbon atoms. 3 to 7 carbon atoms. These monosaccharides contain single polyhydroxyaldehydes and single polyhydroxy ketones are present. Then these monosaccharides are classified into different groups based on different aspects. So first one, so based on the functional groups, that means what type of functional groups are present in monosaccharides. These are classified into two groups. So first one, aldose sugars. Then second one is a keto sugars. Aldose sugars and keto sugars. So based on the functional groups, so what type of functional groups are present in monosaccharides? So these are classified into two sugars, aldose sugar, then ketose sugar. So first one coming to aldose sugar. So generally in aldose sugar, functional group is a aldehyde group is present. So functional group is a aldehyde group. So that's why this is known as a aldose sugars. So examples are glucose, then glyceroldehyde 3-phosphate. That means glyceroldehyde. Then erythrose. Then ribose. So examples are glucose, glyceroldehyde, erythrose and ribose. So among this one, glucose is a most common. Among this one, glucose is most common. That's why glucose is known as a universal sugar. Glucose is known as a universal sugar. Then examples of these are all those sugars, grape sugar. grape sugar, then corn sugar, then dextrose. So main examples of these aldose sugars are glucose, glyceraldehyde, erythrose and ribose. Among this one glucose sugar are most common. This known as a universal sugar. So these are grape sugar, corn sugar and dextrose. So next one, ketose sugars. So in ketose sugars, the main functional group is a ketone group is present. That's why this is known as a ketose sugars. Examples: fructose, then ribulose, xylulose, pseudohexulose, then DHAP, dihydroxyacetone phosphate. So these are the examples of ketose sugars: fructose, ribulose, ribose is a aldose sugar, then ribulose, xylulose, pseudohexulose and DHAP. Among this one is a fructose is a most common. So fructose sugars are fruit sugar, then labulose, fruit sugar and labulose. So fructose sugar, naturally occurring sweetest sugar is a fruit sugar. That means fructose sugars. Next one, based on number of carbon atoms, these monosaccharides are trioses, then tetroses, then pentoses, then hexoses, then heptoses. So based on the number of carbon atoms are present in these monosaccharides, these are trioses, tetroses, pentoses, hexoses and heptoses. So in trioses, 3 carbon atoms are present. So these are the 3 carbon compounds. Examples: glyceraldehyde, 3-phosphate, then DHAP, dihydroxyacetone phosphate. G3P is a aldose sugar, then DHAP is a ketose sugar. Next one, tetroses. These are the 4 carbon compounds. Example: erythrose, 4-phosphate and some trioses. Next one, pentose. It is a 5 carbon compounds. So ribose, then ribulose, xylulose, etc. 5 carbon compounds. Next one, hexoses, 6 carbon compounds. So these are glucose, then fructose, etc. So glucose and fructose, 6 carbon compounds. Next one, heptoses, 7 carbon compounds. So these are pseudo, heptose, etc. So based on number of carbon atoms, trioses 3 carbon compounds, then tetroses 4 carbon compounds, then pentoses 5 carbon compounds, then hexoses. So 6 carbon compounds, then heptoses 7 carbon compounds, pseudo, heptulose, etc. Then these pentoses and hexoses can have the open form structure, then ring form of the structure. So open form and ring form of the structure. So pentose and hexoses can show the open form of structure, then ring form of the structure. So open form of the structure. So this is known as a open form of the structure. Then ring form of the structure. Ring form of the structure is a 2 types. So it is a pyranose, then uranose. Pyranose and uranose. So ring form of structure is a pyranose and uranose. So these pyranose can have the hexagonal shape. Hexagonal shape. These have the pentagonal shape. Hexagonal shape and pentagonal shape. So 1, 2, 3, 4, 5. So 5 corners. So 5 corners represent 5 So these are 5 corners represents 5 carbon atoms along with 1 oxygen. 1 oxygen atom. This type of structure is known as a pyranose ring form of structure. So ring form of structure is a 2 types of pyranose and uranose. So pyranose generally have the hexagonal shape. This contains the 5 carbon, then 1 oxygen atom. So 5 corners. So these are 5 corners represents the 5 carbon atoms. So next one, uranose. This shows the pentagonal shape. So 4 corners. 4 corners represents 4 carbon atoms. So 4 corners represents the 4 carbon atoms. Then 1 oxygen atom. So H, OH, H, OH, H, OH, then H, OH, then CH2, OH. It is a 5th carbon condition. So this contains the 4 carbons, then 1 oxygen atom. This type of ring form is known as a uranose ring form. Next one. Oligosaccharides. Oligosaccharides. So oligo means few. Oligosaccharides means few. Few monosaccharides are present. So generally in monosaccharide, single monomer is present. Simplest unit is present. In oligosaccharides, some number of monosaccharides are present. So approximately 2 to 9 number of monosaccharides are present. So generally these monosaccharides are crystalline condition, water soluble condition, sweet taste in condition. So 2 to 9 monosaccharides are joined in oligosaccharides. These 2 to 9 monosaccharides are joined by the bond, glycosidic bond. So bond is a glycosidic bond. So monosaccharides are joined one with each other by the glycosidic bond. So glycosidic bond formed between 2 monosaccharides. Among the monosaccharides between the So these are carbon atoms, carboxylic group and alcoholic group. That means hydroxyl group, alcoholic group. So these known as a polymerization. That means condensation. In oligosaccharides, monosaccharides are joining up by the glycosidic bond. Glycosidic bond formed between two monosaccharides between the groups of CO group, then alcoholic hydroxyl group by the elimination of water molecule. That means dehydration. Then by the hydrolysis of these oligosaccharides will give the same type of monosaccharides or different types of monosaccharides. So these oligosaccharides are generally water soluble condition, then sweet taste condition. Then based on the number of monosaccharides are present in these oligosaccharides are classified into three classes. So first one is a disaccharides, then trisaccharides, then tetrasaccharides. So disaccharides, trisaccharides, then tetrasaccharides. So first one coming to disaccharides. So in disaccharides, two monosaccharide units are present. Is known as a disaccharides. So best examples are lactose, then maltose, then sucrose, then trehalose is examples for a disaccharide. Lactose, maltose, sucrose and trehalose. Then trisaccharide, three monosaccharide units are present. Example raffinose. Then tetrasaccharide, four monosaccharide units are present. Example stachyose is example. So first one coming to lactose. So lactose is a disaccharide. That means two monosaccharide units are present. This is also known as a milk sugar. This is also known as a milk sugar. This milk sugar generally present in milk of the mammals. Milk of mammals. By the hydrolysis of this lactose sugar will give the one alpha D glucose, then one beta D glucose, alpha D glucose, beta D galactose. By the hydrolysis of this lactose will give the alpha D glucose, then beta D galactose. So in lactose, two monosaccharide sugars are present. So first one is a glucose sugar, second one is a galactose sugar. These two are joining by the glycosidic bond. So these are beta 1,4 glycosidic bond is present. That means first carbon of one monosaccharide, then fourth carbon of second monosaccharide by the elimination of water molecule. That means H2O. So lactose generally known as a milk sugar. These are present in milk of the mammals. So these contains the two monosaccharide units. So alpha D glucose, beta D galactose. By the hydrolysis, it will give the alpha D glucose and beta D galactose. These two are joining by the glycosidic bond, beta 1,4 glycosidic bond by the elimination of one water molecule. Next one, maltose. So maltose also known as a malt sugar. Maltose also known as a malt sugar. These are generally present in germinating seeds. These are generally present in germinating seeds. So these also formed by hydrolysis of starch. These also formed by the hydrolysis of starch. So maltose sugar generally present in germinating seeds. It will also formed by the hydrolysis of starch. So by the hydrolysis of maltose, we'll give the two types of glucose units. These are 2 alpha glucose unit. Same glucose units are present. So these are 2 alpha glucose units are joining by glycosidic bond. So alpha 1,4 glycosidic bond. So by the elimination of water molecule, by the elimination of one water molecule, maltose sugar. Then cellulose. So cellulose is a similar to maltose, but cellulose formed by the hydrolysis of cellulose. By the hydrolysis of cellulose, it is similar to maltose, but in this beta 1,4 glycosidic bond is present. It is similar to lactose sugar. So cellulose formed by the hydrolysis of cellulose. It is similar to maltose, but it shows the glycosidic linkage type is a beta 1,4 linkage. Next one, isomaltose. This isomaltose also similar to maltose, but it shows the alpha 1,4 glycosidic linkage. In maltose, alpha 1,4 glycosidic linkage is present, but in isomaltose, alpha 1,6 glycosidic bond type is present. So next one, sucrose. Sucrose disaccharide. It is a water. So sucrose is a disaccharide. It is a non-reducing sugar. Sucrose is a disaccharide, non-reducing sugar. Then lactose, maltose are the reducing sugars. Then sucrose is a non-reducing sugar. So widely distributed sugar in plants are the sucrose. So commercially this obtained from the sugarcane plants. Sugarcane, then sugarcane and sugarcane. So we can commercially obtain from the plants of sugarcane and sugar beet. Then sucrose contains 2 monosaccharide units. So by the hydrolysis, this sucrose will give the alpha D glucose, then beta D fructose. So by the hydrolysis, this sucrose will give the alpha D glucose, then beta D fructose. So in respiration before glycolysis, this sucrose undergoes to hydrolysis by the enzyme invertase. Then split into glucose and fructose. Then glucose and fructose directly participate in glycolysis process. So this glucose and fructose are joined by the glycosidic bond. Type is a alpha 1,2 glycosidic bond. So by the elimination of one water molecule. So glycosidic bond type is a alpha 1,2 type of a glycosidic bond. Sucrose is a non-reducing sugar. Then 3 halose. So generally 3 halose is present in insects and fungi. In insects and fungi, 3 halose is present. So in this 2 alpha glucose units are present. So by the hydrolysis, this 3 halose gives 2 alpha glucose units. Then glycosidic bond type is alpha 1,1 type bond is present. So alpha 1,1 type bond is present. Next one, raffinose. So raffinose is a trisaccharide. That means 3 monosaccharide units are present. So these are D glucose, then D galactose, then D fructose. So glucose, galactose and fructose, three monosaccharide units are present. These are joined by the glycosidic bonds to glycosidic bonds, then elimination of two water molecules. So these are the oligosaccharides. So next one coming to polysaccharides. So next one polysaccharides. So polysaccharides are polymers of monosaccharides. So polysaccharides are polymer of monosaccharides. So more number of monosaccharide units are present in polysaccharides. That's why monosaccharides are building block for this polysaccharides. These all monosaccharides are joining by the glycosidic bond. So this is known as a polysaccharides. Then by joining these monosaccharides form a long chain of a polysaccharide. This long chain of polysaccharide is a linear condition. So linear condition or a branched condition. So linear or a branched. So polysaccharide chain is a linear straight line condition or a branched in a condition. So it has a left end and a right end. So left end is known as a non-reducing end, then right end is known as a reducing end. So left end is known as a non-reducing end, then a right end is known as a reducing end. Then these polysaccharides are two types of homopolysaccharides, then heteropolysaccharides. So homopolysaccharides, then heteropolysaccharides. Homopolysaccharides contains the same type of or one type of the monosaccharides is known as a homopolysaccharides. Example: starch, glycogen, cellulose, inulin, callose. So these are the examples. Maybe so these are the examples of homopolysaccharide. Homopolysaccharides contains only one type of monosaccharides. So examples of starch, glycogen, cellulose, inulin and callose. So chitin is a complex of polysaccharides, but only one type of glucose units are present. So first one starch. So starch is a widely distributed stored material in a plant kingdom. This starch is synthesized through the process of photosynthesis. Then starch contains the same type of monosaccharides. These are glucose. So starch is a mixture of amylose and amylopectin. So amylase is a linear condition, so without branches. Then amylopectin is a branched condition, so ratio is a 1:4. So starch is a mixture of amylose and amylopectin. Ratio is 1:4. So without branches, then with branches. Then amylose generally 250 to 500 monosaccharides are present in amylose. So amylose contains 250 to 500 monosaccharide units. Then amylopectin contains so over 1000 monosaccharide units are present. Over 1000 monosaccharide units. Then partial hydrolysis of these starch will give the dextrin. So partial hydrolysis of starch will give the dextrin. So in starch, monosaccharides are present. These monosaccharides are joined by the glycosidic bond. So here glucose units are present. So alpha glucose, alpha D glucose units are present. So polysaccharide, the bond is a glycosidic bond. So alpha 1 comma 4 type of glycosidic bond. That means first carbon of one monosaccharide, then fourth carbon of one monosaccharide, then by the elimination of water molecule. So starch. In starch, glycosidic bond type is a alpha 1 comma 4 type. By the partial oxidation, that means the glycolysis of this starch will give the dextrin. Next one is a glycogen. So glycogen is a reserved food material. Glycogen is a reserved food material in animals. That's why this known as a animal starch. So this known as a animal starch. So glycogen is a reserved food materials in animals. That's why this known as a animal starch. Then glycogen generally stored as a colloidal complex of structures in liver and stomach. Liver and stomach. So it is a branched condition. It is a branched condition, but extensively branched than the amylopectin. In starch, amylopectin is a branched condition. That means helical branches are present in starch. Due to these helical branches, these starch can show the I2 molecule. That means iodine molecule. That's why it will give the blue color with the iodine. It will give the blue color with the iodine. So starch shows the helical branching condition. Due to this helical branching, it can stores or binds the iodine molecule. It will give the blue color with the iodine. That means in iodine test. Next one, this glycogen is extensively branched than the amylopectin. There. So glycogen is a homopolysaccharides. That means contain the same type of monosaccharides. So in glycogen, same type of glycogen glucose units are present. These are joining by the glycosidic bond. So by the elimination of water molecule, that means dehydration. So next one coming to cellulose. So cellulose is a like a reserved food material in plants. So generally plant cell wall composed by the cellulose. Plant cell wall composed by the cellulose. Cellulose is a most abundant carbohydrate in plant kingdom. Most abundant material in plant kingdom. So cellulose is a most abundant compound in plant kingdom. Plant cell wall composed by the cellulose. Cellulose is a highly resistant to hydrolysis. That's why plant cell wall is a composed by the cellulose. In cellulose also glucose unit, same type of glucose units are present, like alpha D glucose unit, then beta D glucose unit. But in cellulose, so beta D glucose units are present. So in starch, alpha D glucose units are present, but in cellulose, beta D glucose units are present. These glucose units are joined by glycosidic bond. The type is beta 1 comma 4 glycosidic bond. Beta 1 comma 4 glycosidic bond. So if branched condition, so CH2. So it is a branched condition. So linear condition, then branched condition. So generally cellulose is not shows the helical branching condition. That's why it cannot bind the I2 molecule. So it will not give the blue in color. It may give the red color. may give the red color with the iodine. So cellulose is a most abundant compound in a plant kingdom. Plant cell wall composed by the cellulose. There in cellulose, same type of monosaccharides are present. That's why it is a homopolysaccharide. So this contains beta D glucose units. These beta D glucose units are joining by glycosidic bond. The bond type is a beta 1,4 type with the elimination of water molecule. This cellulose is a highly resistant to hydrolysis. That's why plant cell wall composed by it not shows the helical branching condition. That's why it cannot hold the I2 molecule. So that's why it will not give the blue color with the iodine. It will give the red color with the iodine. So next one, inulin. So inulin is a homopolymer. Inulin is a homopolymer. Glucose units are present. So these are also known as a fructons. So inulin is a polymer of a fructose. Inulin is a polymer of a fructose. These are present in roots. Then tubers of a dahlia. Roots and tubers of a dahlia. But in human body there is no metabolism for this inulin. That means inulin not metabolized in human body. So these will filter by the kidneys. So that's why this inulin will be useful to check the kidney function. This is known as a glomerular function. Glomerular filtration is known as a glomerular filtration. So inulin will be useful to check the kidney function. Why? Because there is no metabolism in a human body for inulin. So it will filter by the kidneys. So it will be useful to check the kidney function. This is known as a glomerular filtration. Next one, so chitin. So generally chitin is a complex polysaccharide, but it contains the same type of glucose units. So generally fungal cell wall and exoskeleton of invertebrates. So fungal cell wall, then exoskeleton of invertebrates. Arthropods. Invertebrates are composed by the chitin. So in chitin only one type of glucose units are present. So these are the NAG, N-acetyl glucosamine. N-acetyl glucosamine. N-acetyl glucosamine is a modified sugar, nitrogen containing modified sugar. So NAG, N-acetyl glucosamine. So chitin contains the same type of monosaccharides but modified monosaccharides. These are NAG, N-acetyl glucosamine. So nitrogen containing glucose sugar. So it is a homopolymer. So fungal cell wall, exoskeleton of invertebrates, arthropods, and these are chitin. Next one, heteropolysaccharides. Heteropolysaccharides. These heteropolysaccharides contains the two or more different types of monosaccharides. So heteropolysaccharides contain two or more number of different types of monosaccharides. So this is known as heteropolysaccharides. Examples agar, peptidoglycan, then pectin, heparin. Examples are heterosaccharides. So agar, peptidoglycans, then pectin and heparin. Then agar and agar. So agar and agar is an industrial product. So we can obtain this agar and agar by the algal members, rhodophyceae members, Xeridium and Gracilia. Next one, peptidoglycan, bacterial cell wall composed by peptidoglycans. So this peptidoglycan contains the two types of sugars. So these are NAM and NAG. So N-acetyl muramic acid, N-acetyl glucosamine, N-acetyl muramic acid, N-acetyl glucosamine. In chitin, only one type of sugar, N-acetyl glucosamine is present, but in peptidoglycans, bacterial cell wall, two types of modified sugars, N-acetyl muramic acid, N-acetyl glucosamine. Then pectin. So generally fungal cell wall composed by cellulose, pectin, algin, etc. So chlorophyceae members. Inner cell wall composed by the cellulose, outer cell wall composed by the algin. Then rhodophyceae members, inner cell wall cellulose, then outer cell wall is pectin condition. Some biophyceae members. So these are the heteropolysaccharides.